All right. So, hi everyone, and welcome to this week's Geotopics seminar. Today we have special guest Dr. Ryan Moyer joining us from the Florida Fish and Wildlife Conservation Commission, the FWC. Dr. Moyer received his Ph.D. in geological sciences from the Ohio State University, Columbus, Ohio. After his doctoral degree, he was awarded as a Mendenhall Postdoctoral Research Fellow at the USGS Center for Coastal and Marine Science in St. Petersburg, Florida. He currently leads the Coastal Wetlands Research Program at FWC's Fish and Wildlife Research Institute in St. Petersburg, Florida. His research is broadly focused on understanding carbon flux and burial in coastal ecosystems, ocean and coastal acidification in estuarine and reef habitats, reef geology and biogeochemistry, and mapping and monitoring of our coastal habitats. All in all, Ryan is a coastal habitat expert in all fields. And today he's going to be talking to us about chasing carbon. So without further ado, thank you so much, Ryan, for being here, and I'll let you take it away.
All right, thanks for that really nice introduction. But Colleen had asked me if I had any awards or anything significant to report, and I'm vying for worst Geotopics seminar, so I'll be happy to take that award at the end of this lecture, but we'll see how this goes. See if I can even advance my slides. All right, so just a brief outline of what I'd like to discuss. I went very broad when I wrote this abstract for the talk. So the talk is going to be very, very broad, and I won't have time to focus on too much detail on anything. We're going to cover a lot of ground here over the next 45 minutes or so. But I'll give a brief background on carbon at the coast and how it cycles, and global carbon cycles in space and time. I will talk a little bit about some of the tools that we use to characterize and trace carbon, so isotopes, and how to use them. I'll then go into some practical research examples and characterizing rivers, characterizing carbon in rivers and coastal environments. Then looking at proxies of coastal carbon flux in coral skeletons, what to do when things don't always work. And then looking at what I spend a lot of my time thinking about now, which is carbon sinks in mangrove-dominated coastal systems. I'll also touch towards the end on implications for management and talking about multiple stressors and how that could potentially impact carbon cycling in mangrove systems. Some implications for blue carbon, which is an emerging buzzword, or has been a buzzword for quite some time now. And a small example of how good management can have implications for carbon cycling, and you can actually see that in some data with some management. So it is fairly ambitious, and that's the ground that I'm going to try to cover for the rest of the talk. So I'd like to start with this slide. This is a fairly old representation of the global carbon cycle, and it says Simplified Global Carbon Cycle. Again, I don't really have time to go through all the boxes and bits and pieces, but it's important to realize that there are some stable pools of carbon that are listed here in the parentheses, and then there are these reservoirs of carbon that actively cycle through on various temporal and spatial scales within the system. One thing to point out on this image is, of course, and this shouldn't be news to anyone in 2021, but there's this large sort of carbon cycle experiment going on where we've successfully tapped into a fairly geologically stable pool, this fossil pool of carbon, and are now just emitting that at unprecedented rates into the atmosphere. So that's one thing that stands out, and I don't know, I'm mousing around right now. I don't know if you can actually see my mouse as I'm using it as a pointer.
Yeah, we could see the mouse.
Oh, perfect. Okay, so we've got this this fossil fuel largely pool, but also land use change and cement production on here. And the other thing of note is that there's really this distinct line, and there's things that happen on land, and there's things that happen in the ocean, but this is the open ocean. And there's really no room for the coast here. And that's a fairly big, well, it's not even a question mark on this carbon cycle. If you just refer to this, you would think that it's all done and dusted. I want to come back to this piece because this transference of carbon from the land to the coastal ocean is rather important and a fairly unconstrained portion of the global carbon cycle. But I want to first take a brief step back in time and talk about what happens when you disturb the carbon cycle, such as we're currently doing with the burning of fossil fuels, land use change, and all of those things. So we've learned that when you have these large carbon cycle perturbation events, and this is from a review paper by Berbel Honish and others that many of you are probably familiar with. Looking at this is called the Geologic Record of Ocean Acidification, and what a team of folks, and I was one of those, that's a preview of the next slide, did was sort of rank geologic carbon cycle perturbation events that have been reconstructed through various proxies, and just kind of rank them in terms of similarity between today's modern carbon cycle perturbation events with the influx of greenhouse gases, and try to come up with a easy to understand ranking scheme, with yellow being the most dissimilar and red being the most similar. The PETM is really the only thing that stands out as the most similar, but whether these events are similar or not, if you look at, and the mixture of yellow and orange is sort of, moderately similar, but still kind of dissimilar. If you look at these, many of these large carbon cycle perturbation events, whether they're similar or not, they spell bad news for coastal ecosystems. And this is just the volume of reefs over these same timescales. And when you have many of these large events, you also have degradation of coastal ecosystems, most noticed in reef building and reef production. And that's one of the easier things that remains in the fossil record. There are other implications for other habitats, but they don't preserve as well. So that's just some geologic and historical context, but we'll bring it back to the modern. And this is, so I started with the simplified global carbon cycle or global carbon budget. And this is the latest and greatest version of that. This is from a paper, I think it was just called the 2020 Global Carbon Budget. And that came out last year. And this is a nice representation. It's got many of the same pools and fluxes that you saw on the previous carbon cycle diagram, but I did mention that there was the lack of sort of any conveyance of carbon from land to the coastal ocean. And in fact, the coast wasn't represented at all. If we zoom in on this piece, you'll notice that there is a budget imbalance here. And that's 0.1 gigatons of carbon, which is not an insignificant amount of carbon. That's quite a lot. And much of that imbalance has to do with these two bits of the budget right here, which is the river term and coasts and coastal ecosystems. And so, and in fact, even with our most sophisticated carbon cycle models, usually we have a really sophisticated land model, we have a really sophisticated ocean carbon model, and then there is this funny thing called the river term that kind of tries to make up for some of this difference and this budget imbalance, even in some of our most sophisticated models. So rivers and the coast are a fairly, to this day, remain a fairly unconstrained portion of the global carbon budget. So it's not unreasonable to spend some time thinking about carbon fluxes and carbon cycling in this part of the world. And so this is a diagram that you may not be able to read. And this is, trust me, this is high resolution, but it's from an older paper. And, you know, those little circles basically that just indicated carbon cycling within the coast or rivers didn't really do it justice, because if you zoom in, the carbon cycle within aquatic systems is fairly complicated. And again, I don't have time to run through all of the pieces here, but there is photosynthesis, there's respiration, there's autochthonous inputs, there's allochthonous inputs, there's inorganic carbon, there's organic carbon, and that's in both particulate forms. Nutrients play a role into the system, even though they're not part of really the carbon cycle itself. And there's a whole other, nitrogen cycle going on here. So it's really complicated. And then all of this gets, if you can read down here, it says export to downriver and marine environments. And yet there seems to be a mismatch. And one of my starting points for all of this was this paper from the late 90s by Hedges, Kyle, and Benner. What happens to terrestrial organic matter in the coastal ocean? And again, they lay out the problem. For a long time, there has been sort of a mismatch. If you just add up all the carbon that comes out of rivers, it can account for most of the organic carbon that is in the ocean, in the sediments. And yet, if you start to look at the character of the carbon in the ocean, it doesn't seem to be terrestrially sourced. So part of this, this imbalance in the carbon budget also has to do with some of the biogeochemical alterations or mechanisms that are going on along the coast. And so this is a student-led proceedings, and I'm sure there are a few students on here. A lesson for the students is don't be afraid to look into older literature. And there's still a lot of good things and a lot of useful things in older literature that you can find and are still relevant to many of today's modern problems. So when we think about the coast, and especially tropical and subtropical systems, many of these they involve estuaries, and many of these estuaries are lined by these intense biogeochemical reactors, which are coastal wetlands. And so a lot of the transformation that we see along the coast can probably be attributed to things that go on in the estuary, and especially the wetlands within estuaries. And there's a need to account for both photosynthesis and respiration. So this is driven by biological processes. But then there is a whole host of transformation and even burial. So there's a lot of sequestration of carbon that happens in these systems too. And some of that carbon that is being brought by rivers to the coast may just be getting trapped before it makes its way to the coastal ocean. Some of it may be transformed into other forms. And again, there's a lot of, even this simplified diagram doesn't really do it justice of all the, fine scale biogeochemical processes that are happening that transform carbon. The burial component also gets interesting because some of it can be sequestered long-term, and we've dated peat sequences in Florida that are, 8,000 years old of continuous organic carbon-rich sediments. That has management implications, which I'd like to get to hopefully by the end of the talk, if I'm still making sense at that point. Because this burial component leads to long-term sequestration of atmospheric CO2 as plants are drawing that out of the atmosphere for photosynthesis. And so this has led to an extreme interest from the management community on these so-called blue carbon habitats. And most of this, and I'll show some actual examples of this from some of my work in Tampa Bay, most of this sequestered carbon is sequestered in the soil, in the peat. So it's sedimentary carbon. And while there are large reservoirs of above ground carbon and below ground biomass, most of the carbon that is sequestered by these systems is stored, if undisturbed, for thousands of years before it potentially gets remineralized and exported to the coastal ocean in a different form. And so the three real dominant systems here that most people talk about are subtropical tidal marshes, tropical and subtropical mangrove forests, and tropical subtropical seagrass beds. So I'll try to loop back to that. Oh, and that's just the transformations and the potential export in a different form going to the coastal ocean. But not all blue carbon is good carbon. You know, we call these so-called estuarine habitats blue carbon habitats, and mostly that's brown sedimentary carbon. It's you know, it's organic rich peat. The actual blue carbon really is the exchange of CO2 with the surface ocean in coastal systems, which is the largest sort of place that atmospheric CO2 can go, is the surface ocean. And when you do that, and you put CO2 into the system, I probably don't have to explain the carbonate system or the inorganic carbon system to most of you, but the system is sort of driven in one direction or the other by pH in the system. And at lower pHs, you have more carbonic acid or vice versa. And the system is driven this way. Higher pHs, you have carbonate. When you have lots of carbonate and you have free calcium in the ocean, you can have calcification by marine calcifiers, such as our friends, the corals here. But if you drive the system the other way, that can promote dissolution of carbonate sediments. And so this is a sequestration of atmospheric carbon by a marine system, but if it's driving the system towards dissolution from a habitat management or an ecosystem perspective, that is not necessarily a good form of blue carbon. So that is something to consider as well. And then of course, we've all heard this termed as ocean acidification, and that's just increasing atmospheric CO2, leads to, among other things, lower pH, which promotes dissolution. It also lowers the aragonite saturation state, which just inhibits calcification within the calcifiers. So not only are the sediments dissolving, but the calcifiers are less able to do what they do best. Okay, and then if you want to start to characterize and start to understand all of these important transformations and start to figure out where's the carbon coming from, where it's going, how is it being transformed, you need some other tools other than just counting or accounting for the carbon in coastal systems. And that's where our friends, the isotopes, come in. I cannot take credit for the picture at the upper left. I egregiously stole this from the internet, and I honestly don't remember where I got it from or what the attributable source is, but it is amazing. And I'm sure Peter Swart is cringing right now, but this is a fun diagram that I use to teach students about how isotopes work and, you know, sort of relative isotope ratios in various carbon pools within the terrestrial and aquatic environment. But if we look at the range of carbon-13 in aquatic environments, and this is specifically at the bottom right, this is specifically sort of a range of stable carbon isotope ratios in salt marshes and various forms of carbon in salt marshes. You can see that different things exhibit a different range of isotope ratios, and that's can help tease out some of these processes and help you also source where some of that carbon is coming from. So here's just an example on the left of how carbon-13 can be used or has been used to look at the increasing influence of corn in the diet of Native Americans over time, and corn being a C4 plant versus C3 plants, which have very different isotopic signatures. You can see the increase in abundance and a shift towards more positive stable isotope values, stable carbon isotope values, indicating an increase in the percentage of corn in diets. If you have two isotopes, the tool becomes even more powerful. This is now stable carbon-13 and sulfur isotopes on a property plot here. And what this is doing is tracing Spartina mussel body tissue that was collected along a transect from a salt marsh, open water. And you can see the sources and the isotope ratios of the sources being constrained organic matter from the Spartina, the marsh grass itself, planktonic organic matter, and then upland plant isotopic ranges. You see they all occupy a different range, and that the organisms collected along this transect, and the stations are numbered 1 from closest to the marsh through 9 to the open ocean. And the data fall out nicely. Those that are closest to the marsh seem to be acquiring the carbon mostly from the marsh. And then as you get closer to the open water, you start to get an increasing influence of plankton. Meanwhile, upland plants possibly for some of these mussels, but definitely not the ones that are out towards the open ocean. So you can use isotopes as a powerful tool to sort of understand where your carbon's coming from and start to try to make sense of all these interesting transformations that occur along the coast. This also works, by the way, since I mentioned using two isotopes, and that's what I'll show you coming up in a few slides. I used carbon-14, and so I used two different isotopic forms, a stable form of carbon, carbon-13 isotope ratios, and then a radioactive form of carbon, carbon-14. And so this also works for radiocarbon. Radiocarbon's also present in all of these same sort of boxes that we saw with the various global carbon budgets that I presented. And because radiocarbon decays over time, it can give you a sense of the age of your material. So we take all of this and we put it together. And this is really where I got my research start into all of this, was trying to make sense of all of this in a river system, and then looking to corals as sort of recorders of all of the sort of integrators of this complex carbon cycling and these carbon discharges from land to the coastal ocean. And so this is just a conceptual diagram of some of the work that I did in this area, looking, and there's a seasonal component. So along the bottom, you've got the dry season, and this is for two catchments in Puerto Rico, and they have very strong wet and dry seasons. And the rivers are barely discharging during the dry season, but they are flowing pretty tremendously during the wet seasons. I've got the expected stable carbon isotope ratios and the expected C14 values. These are just relative ages, relatively younger or older. And there's a difference here in the expected carbon-13 values because of the land use of the region. And Puerto Rico also has a pretty pronounced land use history, where at one time most of the vegetation on the island was cut down for sugarcane, which is a C4 plant. And over time it has been reforested largely by C3 plants. And so those two plants having different stable isotopic ratios, you might expect that in the plants and the soils within each of those catchments. That material then gets into the river, undergoes some fractionation, but you still have values that are reflective of the land sources within the river. That then moves out to the coastal ocean, gets diluted by seawater, and then eventually corals draw from the dissolved inorganic carbon pool for calcification. And so if they're dissolving from DIC, which is being oxidized as it's making its way from the river to the coast, then if you can find corals close enough to your river, you may have a record of carbon flux coming out of that river. And so that was something that I explored largely for my dissertation work a long time ago. But the first step there was just making sure, you always have to verify what your conceptual model and your hypotheses, and make sure what you think is happening is happening. So the first step is to constrain the sources of DOC. And I looked at the particular organic carbon, dissolved organic carbon, and dissolved inorganic carbon. Don't have time. I'm going to set the particulate aside for a moment and or for this presentation. I'll just talk about DOC and also the DIC, since that's what ultimately makes its way to the coral. And on all of these plots, you will see plots of carbon-13 versus carbon-14, and the boxes indicate sort of expected ranges of those sources based on largely published literature values. When you see the river in DOC, that's all based on published values, and then the dots and the triangles are the actual data. The open circles are dry season. The closed symbols are wet season. And you can see that we were able to constrain fairly well the sources of river in DOC, and they all fell within the expected ranges of sources. And to simplify things, I used a 3N member mixing model. So even though there are potentially more sources, we sort of tried to boil that down to the three major sources of river and DOC. C3 plants having, being the largest, soils being the next, or actually equal component, agricultural soils, which are relatively younger, and then pre-agricultural soils, which are relatively older and have lower carbon-14 values. Then we look at what's coming out of the river and try to, that all gets distilled basically into one box here on this plot, and trying to constrain the marine and estuarine DOC that was measured in the nearshore. That was a little bit harder to constrain, and some of these things fell slightly outside the range. I was also a little bit conservative in how I estimated these values, because I threw out some of these larger outliers from the river and DOC as potential sources as well. So that may have helped constrain it a little bit better, but it was really not surprising that marine DOC is slightly harder to constrain because of the aforementioned mismatch between terrestrial organic carbon and marine organic carbon that we saw. So we did this and characterized this in the watershed and the coastal waters in 2 different seasons, and came up with these sources. And I should also mention, so the average and then the range is also given here for all of these sources. When you get to the coast, it's largely a balance between what's coming out of the river and then marine primary production. One thing that was sort of a very interesting find, though, was when we were looking at the age and the carbon-14 ages of the dissolved organic carbon coming out of the rivers in Puerto Rico. And this is, one of those questions about what happens to terrestrial organic matter in the coastal ocean. Terrestrial organic matter in rivers is usually fairly young. It's usually a couple of 100 years, radiocarbon years old, to usually up to about 1,000 years old at maximum. And most systems are only a few hundred years old organic carbon. However, when we measured, and we were able to do this multiple times in multiple locations, but again, there is high variability, and it also depends when and where you sample, because this was not consistently across the board older carbon. But we were very surprised to get some very, very old radiocarbon ages associated with DOC collected from directly from the river. And this is something that, if you look at this with and compare it with other river systems, you really don't see that. Again, most of them tend to be within about 500 years old. And in fact, that's what we found in our non-agricultural river catchment in Puerto Rico, but with the agricultural one. What we think is happening here is that this has just been highly altered. It's a highly altered human system. And there are many pipes just coming into and out of irrigation canals. And these pipes drain directly into or from soils, agricultural soils, but there's not much turnover at the depth at which these pipes have been installed. It seems like they were just kind of drilled laterally and put in there as drainage. And so what we think is happening is that much older, deeper soil layers are actually, some of that organic carbon is then getting discharged as the water, as surface waters percolate through and drain out these pipes. And we think that we're, getting a much older direct source of carbon, and then this irrigation canal connects directly to the river. So we think that's what's going on there, and the reason. But this was rather exciting to find very old carbon draining directly to the ocean, because you rarely find that in rivers. And that has been, again, one of those mismatches between terrestrial carbon and oceanic carbon at the coast. So then, because much of the DOC is oxidized and transformed to DIC, and because then, since the corals were the end game here, the DIC is the source upon which corals draw from for calcification. We also had to quantify sources of both riverine and coastal DIC. And so again, our sources were fairly well constrained within the river, and largely a product of mostly atmospheric CO2 and equilibration with atmospheric CO2, which is a little bit more complicated in rivers than it is in the surface ocean, but still largely driven by equilibration with atmospheric CO2. And then riverine DOC and POC was the next largest contributor. Our sources, no surprise, especially in the marine sites, largely derived from the atmosphere. But you can see this gradient between the river and the marine end members, and we did have this gradient where you can actually see, DIC being derived from potentially oxidation of organic matter delivered by the rivers. So you take all of this, you now understand the sources. And then the next question was, can coral skeletons record coastal carbon cycling? And skeletons, and especially isotope proxies and skeletons, coral skeletons, have been used for a whole host of things. And especially with carbon isotopes in coral skeletons, it gets pretty complicated pretty quickly. There are a number of things that you can measure. So I'm sure many of you are familiar with X-radiographs of corals, and that corals grow with these annual growth bands. And basically each couple of a light and dark band represents 1 year of growth. From that, you can get the age of the coral. So you have sort of an independent timeline chronometer from the coral. You can also measure some physiological things that occurred within the coral, like extension, skeletal density, and calcification of the coral. But then more of interest to this talk are some of the things that you can do geochemically with the coral skeleton, including measuring stable isotopes, radiometric isotopes, trace elements, and some folks have looked at organic inclusions that get sort of bound within the inorganic skeletal matrix as a recorder of terrestrial processes in coral skeletons. So I'm going to focus largely on the first three, and I didn't do any work with organic inclusions directly, but they do serve as effective tools. And again, I'm broadly glossing over the details here, but I'd be happy to discuss it with any of you offline if necessary. So, oops, fast forwarded again. We had a system we looked at this in corals in 2 systems in Puerto Rico, and I'm just presenting data from one, from the Fajardo River in eastern Puerto Rico, And this is a situation where you have a small river draining the eastern mountains of Puerto Rico, discharges to the coastal ocean, and there's a reef about 1 kilometer offshore, drilled a coral, obtained a coral core, and looked at, now you're looking at carbon-13 versus carbon-14 in the DIC component of marine and river and waters. And you'll see the relationship, the slope, and there I go again, getting overly clicky. The trackpad on this laptop is not very good. We found this relationship within the water column. And then we also have a similar relationship within the coral skeleton, where when you're having strong river influence, you have lower carbon-13 and lower carbon-14. And when you have no river influence and largely a marine influence during the dry season, you have higher carbon-13 and higher carbon-14. And there is a statistical test to compare slopes. And in fact, these two slopes, even though they look somewhat different, are not statistically different from one another. So that was, rather exciting. And then we confirmed this with a few other proxies. This is now a very busy slide. We've got river discharge in blue, barium calcium ratios, which have been used as a proxy of river discharge. We've got inverted axes for carbon-13 and carbon-14. And again, we expect lower values going up in these inverted axes when you have stronger river discharge and higher carbon fluxes coming out of the river. And in fact, for both a period where we had gauged river discharge, and then just using the barium calcium ratios and the relationship between the two in the younger period of the core, reconstructing river discharge from that barium calcium proxy, where we had that and where we had the two carbon-13 and carbon-14 isotope values, we had a strong river-derived signal. There are, of course, some caveats with that. And that is that it's we think this is DIC that's coming out of the river, and it does serve as sort of an indication of river influx to the marine environment, but it's not fully quantitative. And so that's one limitation of this, is that, in terms of retracing the carbon cycle, it's not quantitative. For that, you need calibration work. And this is just some calibration work that I did with a completely different isotope system. And this is looking at boron isotopes. And I don't know what happened to my axes, because on the right, you should have been seeing the axes for boron isotopes, but they were there this morning. The presentation has eaten them. What you're looking at is measured in situ pH in the blue and the gray, both discrete and autonomous measurements. This is the reconstructed pH from 4 samples within 1 year within the coral skeleton, and this is a bulk sample of the same year. And this is the type of calibration work that it would need to make the carbon proxy a little bit more quantitative. You can see that we've basically recreated, although it's not fully one-to-one, this annual cycle in pH that we get with our proxy. And in fact, our single bulk sample falls squarely in the middle of our reconstructed pH, falls squarely in the middle of our measured pH. That curve, by the way, is also reflective of sort of the curve of expectations for emerging technologies. And that's where especially the carbon isotopes proxies were, and sort of left those in the trough of disillusionment for several reasons. But one of those is because it doesn't work everywhere. It works great in a system where you have a small river that is not swamping the reef, and that there's a coral growing happily 1 kilometer offshore, which is in a very far distance. What do you do in a system like Florida, where you have a very diffuse flow? You have a big Florida Bay here, lots of mangroves lining the system, and then you have lots of human modifications to that freshwater flow. you're not going to, it was hard to tease out a signal in a system where this could work perfectly. You're not going to be able to tease that out in a system that's as diffuse. So that's where the pit of despair, or the pit of disillusionment comes in a little bit with these proxies. There would be a lot more work to make this fully quantitative. Also, to put this in context, the systems that that I was dealing with in Puerto Rico, these are small river systems, which have been historically, not represented in global carbon budgets. But if you look at other small river systems, in terms of discharge and fluxes of carbon, both from the river and out to the coastal ocean, the fluxes are larger by, 2 to 5 times larger, depending on what you're looking at. So these systems are really small potatoes, and I've been spending a lot more time recently working in some other small river systems and estuary systems within Florida, within Southwest Florida, from Tampa Bay down to the Northwestern Everglades. I'm running close to time, so I don't want to belabor the point too much here, but what we're looking at is a conservative mixing curve of dissolved organic carbon within Tampa Bay. And this is the conservative mixing line for the wet season, and there is a very large sink here, and that is largely within the estuarine waters. And that's that mangrove sink that I talked about, that wetland carbon sink that I talked about. So I'm gonna breeze through the rest of these slides fairly quickly. They are fairly light on data. The Puerto Rico story was the larger one, but I will try to breeze through these slides as quickly as I can. So what about carbon in coastal wetlands, and trying to look at some of these stocks and this burial component, this large sink that we see in coastal estuaries. This is a project that we're working on right now, funded by the USDA and a host of, along with a host of collaborators. This is just from a poster that I gave at a recent, again, one of the last conferences I went to before all of this got shut down, at an AGU Chapman conference. But the crux of it is that we're using a number of different tools, including radiocarbon and lead-210, to look at carbon burial on various timescales. And we're also measuring above ground carbon using traditional techniques and through some remote sensing techniques, and also looking at some of the fine scale biogeochemistry to try to understand the fate and transformation of carbon in these systems. And that's really where my research has gone since the Puerto Rico work. And so I've traded coral coring for mangrove coring, but it's still core-based research, and that's important because, again, most of the organic carbon that is sequestered by these systems, that you find in these systems, it's held long-term for thousands of years, and the bulk of it is in both mangroves and salt marshes. And this is from a characterization we did in Tampa Bay. It's below ground. So you still have to do core-based studies. It's just trading corals for peat cores. Both of them present their challenges to collect. And it always involves a slog of heavy gear through adverse conditions. I said I wanted to end by touching on management, so that's my final few slides, and just a few things to consider when we think about management of these systems, because when we get into coastal habitats, and especially with my current agency, FWC, managing these habitats is one of the bigger things we think about. On a positive note, we hear a lot of bad things about climate change and sea level rise, but on a positive note, we've looked at organic carbon burial in the Southwestern Everglades. And what we see is that there has been increasing rates of carbon burial over time. So these are lead-210 ages down core, and then you're looking at rates of carbon burial, and everywhere we've looked, from the 10,000 Islands down to Shark River Valley, the rates of carbon burial are increasing with sea level rise. So that's potentially a good thing from a carbon cycling perspective, and maybe one less thing you have to worry about from a management concern, because you're getting carbon sequestration out of these habitats, and sea level rise makes accommodation space. However, sea level rise isn't the only stressor, and most of these systems, especially in places like Florida, have multiple stressors. And this really is a management concern, because the combined effects of multiple stressors, and this is true whether you're looking at mangroves or reefs or pretty much any coastal system, combined effects of multiple stressors usually lead to ecosystem degradation. And if you decrease the health of your wetland, you fundamentally alter the carbon cycling and carbon storage, and you switch these wetlands from net sinks to net sources. So this is from a proposal that I just put in with colleagues to the Department of Energy to try to do some mathematical modeling and understand that a little bit better. The opportunity with the management, if you can understand those things and if you can promote carbon burial over habitat degradation, is that these systems are effective reservoirs of carbon. And we have proposed this as a driver to incentivize restoration and preservation, because there are emerging voluntary carbon markets. There are mandatory markets in other countries, and we've seen interest in foreign investment of wetland preservation here in the United States. And so we've proposed this as an incentivized driver. So you get carbon cycle benefits out of preserving coastal wetlands, and one drives the other. The last thing I'll touch on, because I'm 2 minutes over time, is that we actually see some benefits in the inorganic carbon system with habitat restoration. And so the story in Tampa Bay is that water quality was severely degraded in the 1970s. Seagrass declined considerably. And over that time, through nitrogen improvements and things like this, water quality has improved, and the seagrass acreage has returned to 1950s levels, which was one time thought impossible. As the seagrass has returned, and you're getting more photosynthesis in the system versus more respiration in the system, it's driving the inorganic carbon system. We see a measurable increase in pH, which is opposite of the ocean acidification trend that is being exhibited in most places. And with effective management of seagrass, we've actually seen an increase in pH and a shift in the inorganic carbon system towards towards net calcification over net dissolution. So there are implications for management, and they tie back to the, you can see the successes in the carbon cycle. So this is just a summary slide of, again, the very broad ground that I've covered. Sources of carbon are large, but poorly constrained. We were able to constrain them in Puerto Rico, but it's a very small, very simple system. We did have evidence that coral skeletons could record coastal carbon dynamics, but it doesn't work everywhere, and it's not fully quantitative. A lot more work needs to go into that. Puerto Rico is a simple system, as I've mentioned, and there's lots of transformations going on, and really wetlands are the place if you want to look for those. So to end, I tried to put acknowledgement slides together, and I had literally 6 slides full of, like a quarter of the length of this talk, just in acknowledgement slides. I have benefited greatly from, and it being, it's Women's History Month in March. So I opted to just pull out a subset of the people responsible for this talk. And really none of this would have been possible without all of the wonderful opportunities I've been given by these fine people. So that's not a misspelling of acknowledgements. That's just who I chose. I am leaving out a lot of people and a lot of funding sources. I'm very grateful for them. I also want to thank the University of Miami, and specifically folks at Rasmus have also been instrumental. Again, they haven't really contributed to much of this work, although I've done some boron measurements with Ali and done some ocean acidification work with Chris. And Peter is the one that got me down the dark road of isotopes to begin with. Pam Reed was on my master's committee. Sam Purkis, I'll just leave it at Sam Purkis. And Bob Ginsberg just gave me some incredible opportunities when I was just starting out in this field that I'm really appreciative of. My new best friend is also Colleen, who invited me for this. So thank you, Colleen, for letting me do this, and I'll take that world's worst trophy now if you've got it. I'll take some questions if I left any time for questions.
Right, shall I, can I start with the first question?
Yeah, Sam.
I mean, first of all, it's a super talk, and my question is a bit tongue-in-cheek because I'm going to play devil's advocate on the blue carbon just for a second. And in the rock record, we rarely find meaningful high TOC deposits in the coastal zone. You don't find a lot of carbon preserved in the coastal zone. It's always in the deep anoxic ocean. I suppose you'd call those dead zones in today's parlance. How do you resolve that paradox with blue carbon? You said that you get 8,000 years of storage, maybe in Tampa mangroves. Is that meaningful on a climate change perspective? Could you reconcile that paradox? What are your thoughts?
That is a wonderful question, Sam, and it's a really good one, and it's I can't really. Yeah, I don't even think you're playing devil's advocate here. I think you're spot on. I think it depends on your time scale. And of course, the blue carbon community is a very management and economically driven community. And so the time scales that they are thinking on are not always the time scales that geologists think on. And that's, even though it was pretty tangential, that's why I tried to wrap in that whole carbon cycle perturbation event slide at the very beginning. I think that perspective is a good one. There's a lot, and that multiple stressor slide that I had, you can convert net sinks to net sources. That's fairly easy to do. And we know that anytime there's been significant changes in climate and or sea level, we have pretty significant upheaval at the coast. And sometimes the coast is at the edge of a continental shelf, and sometimes the coast is, the Keys don't have a coast, for instance. And so there is probably, a huge amount of missing carbon in the fossil record just because of the large amount of upheaval that goes on just over the climate changes we've seen over the place, the changes in sea level we've seen over the Pleistocene, for instance. You're probably losing a lot of that record. However, to a manager, 8,000 years is a long time. And if you can sequester carbon for that long, we probably should be trying to maximize it. I agree with you. There's a huge disparity between the time scales. If that is significant in terms of forcing the climate system, it probably helps ameliorate it some, but I don't think,
Well, I mean, I think you've made the most powerful point that I got is the fact that you've identified a negative feedback to a warming climate. We're so familiar with positive feedbacks, releasing methane from the tundra, releasing methane from the continental shelf. There's always positive feedbacks amplifying the changing climate, but you've identified a negative, even if it's temporary, only measured in millennia, that as you're warming the planet, the coastal zone actually increases its capacity to store carbon, and thereby putting the brakes to a degree, on the warming. That's a really rare thing that you've identified. And even if it's only temporary, you make exactly the right point. We should be doing it anyway.
Yeah, and I mean, there are also caveats to that, and there are some thresholds. So, for instance, they've done studies of how much CO2 increases productivity to a point, and then when CO2 concentrations in the atmosphere exceed a threshold, productivity in plants actually decreases. So you're going to get a less autochthonous production of carbon and less of that sequestration pathway at higher CO2 concentrations. And sea level rise at slow rates, and we're still fairly slow rates, but increasing, creates accommodation space for burial. But again, if those rates get too fast and you start to actually drown your systems in place, then you switch that feedback. And so it's again, the complexities there are, and that's part of what we're doing with that conceptual diagram I showed you of looking at carbon burial on several different timescales. Because even if you look at rates of burial on different timescales, you get different answers down core. And so there's a lot to be teased out there.
All right, I'll ask a question, Ryan.
Thanks, Peter.
Can't let your carbon isotope stuff go uncommented
no.
on. So I'd say 99.9999% of all papers dealing with carbon isotopes in corals basically concentrate on the physiological control of the carbon isotope record in the coral skeleton, which you didn't mention at all. And I'm just wondering how you're unraveling the physiological controls of which 99.99999% of all the papers have focused on, and now all of a sudden it's all about what is being delivered by the rivers.
I think I recall a Swart paper from the 90s that actually opened the doorway for other pathways besides just metabolic control, including DIC. That is a good question. And again, carbon isotopes get extremely complicated. The way that we, the way that I was thinking about that is that's where the addition of the carbon-14 isotope comes in.
Mm-hmm.
Because if it was all metabolic, it should be bomb carbon or relatively young carbon. And we're seeing older carbon come in specifically at times that we know that there's higher river discharge. And again, because we could constrain the DIC isotopically coming out of the river, we could argue this, and I know there probably are some stretches there, but the radiocarbon is really what was meant to sort of isolate from the fully metabolic pathway for carbon isotopes. That's the short answer.
It just seems like, during the summer, which presumably is what the wet season is, that there should be some some overprint of your river signal by the metabolic cycle, and that you should be able to tease it out. Now, I have to confess, I haven't read your landmark papers on the Puerto Rico stuff in detail, but I just wondered if you, when you were doing that, whether you and Andrea, who is also a goddess of physiological control of carbon in coral skeletons, had to, discuss this.
Yeah, and we did. And again, largely our discussions came back to using the radiocarbon as sort of an independent tracer that gives you a non-metabolic signal, basically. And so that's why it's it's a much longer conversation to answer, as I know it's a much longer conversation to answer that question. But there's also upwelling of, there's papers looking at upwelling, so other sources of DIC, and it is difficult. The simple answer is the carbon-14 is what is meant to represent a non-metabolic carbon pathway, and why we chose to do that. And as you're also aware, most of the physiological papers that use carbon-14 just use tracer concentration carbon-14 and don't use natural abundance carbon-14. So.
All right, I'll leave it there. As you said, it's a long conversation.
We can we can spend hours on the phone or Zoom discussing that one because it's a long conversation.
I have one quick question to something that Sam had brought up, and that's the carbon sequestration increasing with sea level rise. I was wondering, is that ability for carbon to be sequestered just increasing because there's more CO2 in the atmosphere, or also in conjunction with the sea level rise creating more accommodation space?
It's a little bit of both. It's a combination of higher rates of productivity. So you're getting more in situ production of autochthonous organic matter. And then also the sea level just creating accommodation space. And that's, you know, one potential mechanism. If you dive into the paper, and again, I glossed over the details, but if you, there's three other mechanisms that we presented in the paper. And in fact, that was not without controversy because we just got a comment that we had to reply to in the journal. And that actually just got accepted, and we just got the proofs yesterday. So look for that. If you're really interested in this story, the paper came out last year. It's Josh, lead author is Josh Breithaupt. And then there is a response and or a comment and a response and that's JGR Biogeosciences. That should be, I think it's available unformatted online already. And then we just got the proof. So probably by next week, it'll be done and dusted in the final format.
Exciting. Well, thank you. If there are no other questions, I know we're just a couple minutes over here. I just want to say thank you so much for giving such a wonderful presentation today and for taking the time to talk to us about your amazing research.
Thank you. Thanks for having me. I wish I was actually back at Rasmus, but thanks anyway.
Thank you, Ryan. You're welcome anytime. You're with us in spirit.
Thanks, Sam.
Thanks, Ryan.
Right.
Thanks, Peter.
Really super.
While I'm thinking about it and while Colleen's still on, is there a way, so I invited some students for extra credit. Is there a way to get a roster or anything of who actually attended?
I don't think it saves on here on who attended, so you may just have to trust them.
Okay. I can do that.
It might in the recording, Colleen. Maybe you should have it might.
Yeah, it may say in the recording. I'll have to check the log.